Hi, I'm Leah Nandudu, a PhD candidate at Cornell University in John Locke Yannick's lab, and today I'll be giving an update and also explaining the concept behind root necrosis imaging, CBSD root necrosis imaging. Now, as previously mentioned by all the earlier presenters in this talk, you've probably understood why it's very important for breeders and pathologists working with cassava brown streak disease to identify sources of resistance in different populations of cassava, and also markers to enable marker-assisted breeding if we are to have the concept of CBSD resistance breeding working out, to get resistant clones to CBSD. Now, it's important to note that right now, to differentiate between severely infected clones from less severely infected clones, we use the 1 to 5 scoring method in the field. This method is applied both on the foliar and the root symptoms, and a score of 1 indicates no observable symptom, while a score of 5 is the highest severity possible on both tissue types. Now, the next slide that I'll show you is from the Cycle 2 population of genomic selection that was planted in 2 locations, Namlonge and Serere, in 2 growing seasons. Now, while fitting mixed models, we use data from the 1 to 5 scoring scale to partition variance that's explained by the genotype, the environment, and genotype by environment interaction. And it's good to highlight that when we use the 1 to 5 scoring scale, most of the proportion of variance that's observed is actually being explained by the genotype, which is very good for breeders. And when we look at genotype by environment interaction and the effects from the environment, they're generally low, with generally low from CBSD 3 and 6 months after planting. But when it comes to CBSD at 12 months after planting, we have 0 genotype by environment interaction in the Cycle 2 population, and a very small proportion of variance being explained by the environment, which is very good for a breeder. But then when we go ahead to look at how much of this variance is due to the genetic effects, right? So we did narrow-sense heritability, and you could see that we generally have low narrow-sense heritability for CBSD foliar and root scores at 3, 6, and 12 months after planting. Now, this means that most of the phenotypic variance we are observing is actually not due to the genetic effects. Now, we went ahead to conduct genome-wide association studies for cassava brown streak disease in the Cycle 2 population, and I'll concentrate on incidence and severity at 12 months after planting, because these generally are from the root necrosis, which is our interest in this moment. Now, you could see that we pick out significant peaks on chromosome 4 for incidence and chromosome 11 and 16 for severity. It's important to note that these genomic regions have been reported in previous studies like those of Kayondo and Morag, but just as the previous researchers reported that there was inconsistency in how SNPs showed up. So SNPs were either inconsistent across years and location. This is the same thing that we're seeing in the Cycle 2 population. And again, then the SNPs in those regions, in these regions associated with CBSD phenotypes, generally explain a small proportion of variance. And so that makes us ask the question, what more can we get from root necrosis? What more information can we get from this? Because we know that when you look at CBSD at 3 and 6 months after planting, which applies to the foliar, there are 2 time points of study. So we do have a lot of information about what happens to the foliar symptoms. But then the root symptoms, which are of economic importance, since root necrosis makes the root unusable, we have very little data since we only have 1 time point on it. So we got thinking that why can't we take images of root necrosis and try to extract different information to help us understand the genetic mechanism of CBSD root necrosis. So as you can see here, like there are different patterns of root necrosis expression, and this is due to a direct impact of the replication of the virus at these different spots in the root. You can see it can either start at the center of the root, or as small lesions or large lesions, or at the edge of the root with different expressions, or just, doesn't have a pattern of expressing. So what we're planning now is to use images to extract information on the percentage of necrosis, which is generally the percentage of the necrotic lesion to the root area. And we also want to do account for the number of lesions and the size of lesions, so we could actually use imaging tools to extract the size of lesions, as you can see in this case, but also the centrality. Now, to explain the concept of the centrality, we determine the center of the root, right? And then from the center, we can determine how far different necrotic lesions are from the center of the root. So if the necrotic lesion starts at the edge of the root, you can determine how far is it from the edge of the root to the center of the root. And if it's the center of the root, then you know that from that necrotic lesion, how far is it from the center of the root, which gives us a picture on virus replication and virus movement. Now, what we've done again, as I highlighted about the cycle 2 population, but just in detail, the cycle 2 population is made up of 464 clones that were planted in Namlong and Serere for 2 years. So far, we have 61,000 images that need to be analyzed. And how these images were collected is we did from each plant in a plot. So a plot is made up of 10 plants. So from each plant, we did randomly collect 5 roots that were sliced at the head, the middle part, and the tail. And we did take pictures at the 3 parts of these roots for root necrosis. Now, an analysis pipeline is at its later stages, so we're about to use the pipeline. And this has been developed by the Makerere University AI lab, headed by Dr. Joyce. Now, what we hope to understand, or information, the expected outcomes we hope from this root necrosis data is, one, we hope to expand CBSD phenotyping, because you can clearly see that the 1 to 5 scoring cell, despite being efficient, it has its own limitations. But also we hope to unravel the genetic mechanisms of CBSD root necrosis, which are very crucial for us in identifying markers and also validating these markers. And also in the end, we hope to at least identify, either resistance loci, loci that are responsible for resistance, or susceptibility loci, information that can be leveraged when breeding for CBSD resistance. I thank you so much for listening to me. If you have any questions or suggestions, you can reach me via email. I shared it earlier in the slide. Thank you so much, and hope to hear from you soon.